be telling you a little bit today about standards and quality control for light microscopy. So as Lionel said, throughout my whole career, I've always been focused on measuring things in images. So not just getting a picture, but actually measuring what's in the images. And we really have to educate people and make sure people using the microscopes understand what needs to be done in order to make sure that those images are quantitative. So I've broken the lecture today up into 7 different sections. So I'm going to talk a little bit about cleaning. That's something I don't enjoy, but it's very important for the microscope. About stability and power of the light source, resolution, field uniformity, so the fact that the intensity of illumination light is not the same across the whole field of view, linearity of detectors, registration of colors for multicolor imaging, and then I'll tell you a little bit about a study on 3D standards and metrics that we're hopefully submitting for publication shortly as part of the Association of Biomolecular Resource Facilities Light Microscopy Research Group. So I'll start with cleaning. So there's a lot of different tools out there for cleaning, and I know one that I learned from the Stowers Institute in Kansas City were these air puffers, which are like a little shaped like a little rocket, so they're kind of fun. And I've put like information on enough information that you should be able to find these objects or these different products around. And we always recommend that people don't use these air canisters because they have solvents in them that can damage the optics on the microscope. Other simple things like Swiffer dusters and sticky mats to keep dust down in the rooms. Non-ammonia based glass cleaners. So we tend to use just regular glass cleaners for most of our cleaning and stay away from harsh solvents. But we buy these products commercially and dilute them about 1 to 5. So 1 bottle lasts for a really long time. We also found these neat little spiral clean room swabs, which are used, I think, more in nanotechnology. And we wrap a little bit of lens tissue around these for doing some real deep cleaning under the dissection microscope. And last is a polymer material called First Contact, where you can actually paint it on kind of like nail polish, let it dry, and then peel it off. And this is really good for really sensitive surfaces like the front glass on a camera and so on, that can be difficult to get at. And I'm happy to share these slides as well after the presentation, and I try to put in lots of links. So all of these text that's in blue here is all linked to the original articles, and I won't go through in detail here, but there's a number of articles here on keeping the microscope clean and the importance of it in different techniques. This third one here, I couldn't find a digital version, so if anybody's interested in that one, I can send it to you directly. And then there's a couple websites here, one out of some of Mike Davidson's work from Florida State University, and then a nice Jove video article as well. So I won't say any more about cleaning, but don't underestimate the importance of that and how important it is to get good quantitative images. So the second one is instant light power and stability, which we've spent quite a bit of time on myself, and there's many new initiatives as well. So we did a study with the ABRF Light Microscopy Research Group quite a few years ago, where we looked at light power and stability. And we looked at different time scales because the stability is important on different time scales depending on your experiment. So if you're taking a single image, you want to make sure that the light's not fluctuating during that time when you're scanning. If you're taking a Z-stack, you want to make sure that it's stable over the minutes that it might take to take a high resolution Z-stack. If you're doing time-lapse, you might be looking for hours. And if you're doing replicates of your experiment over many different days, you want to make sure that when you go back to the microscope each time, the light source is at the same power. So it's really important to actually measure the power with a power meter when you go to the microscope and not just rely on the settings in the software, which are usually just in an arbitrary unit of % power of some sort. So that's really important. And then one of the things we kind of struggle with, and maybe during the Q&A, I'd be happy to chat with people if people have ideas, but we always struggle with what happens when we have a laser that gets replaced, or we replace the bulb on a light source. You know, and the users may not be aware of that, or even if we make them aware of it, do they know how to handle it? And so that kind of comes back to if you measure the power with a power meter every time you go, then you can minimize the impact of those things. So this is, and throughout the presentation, I'm trying to give you lots of tips and tools to use. So this is just a series of different commercial products that are available for measuring laser power. We have the Acceleratus one and the Coherent one. One of the nice things about the Thor Labs one is I believe that you can also use it with oil immersion lenses. And the ones that mount on the stage like a slide are usually a little bit easier to get reproducibility. The Coherent one, you have to kind of mount it in the slide holder and push it in place to hold it tight on the stage. And so sometimes just even going from user to user, you get a lot of variability just because the sensor is not placed in the same location. So what we normally do is we normally take a 10 or 20x dry lens, focus on the sample, and then take the sample off with the power meter on and measure the power there. So even if you're using an immersion lens, if you always have the same power with the 20x lens, then that power will be higher with the higher magnification, but the relative power will be the same. So it's an easier way to measure it than having to have an oil immersion sensor. If you don't have access to a power meter, we did this study with the ABRF that was published back in 2011, and we just used plastic fluorescent slides. So I'm sure all of you have seen these chroma slides when you go to the conferences and so on, and they're usually giving them away for free. And you can put that on the stage and then just image that over time and then measure the intensity. And you can do a fairly small region of interest. You don't really need to take a really large image area. And this works quite well. One thing is you can have some focus drift. So if you're on a laser scanning confocal or a wide field system, you can also in the laser scanning confocal, you can also take an image with the transmitted light detector, and that should be measuring the laser light getting straight through. And so there you can look at the two data sets and see if you're seeing some focus drift. One thing we did find is that these slides are super bright, so we usually mismatch them. So if you want to look at your blue laser and green fluorescence, we tend to put the pink slide on. And if you want to look at red fluorescence, you put the green one on because they're just so bright. It's hard to work with them when their colors are matched. And I think with the red one, we can get all of the lasers except for the 405. So you can actually do 3 or 4 colors and look at the stability of many light sources at once. So this is just an example of some of the data you can get. So on the left side here, we have a 633 laser, which is quite stable. And we measured both the fluorescent light getting to the PMT and the transmitted light. And you can see the scaling here is only 75 to 100%. So it's just a couple of percent variation. But on the same microscope, we looked at the 561 laser, and you can see it was fluctuating by about 5 or 10% in a pretty periodic way. And it turned out to be an issue with the cooling of the laser. And you can imagine if you were doing an experiment and you were collecting a Z-stack, you probably wouldn't notice this. It's just 5% change in intensity. So if we hadn't been doing this standard test, we wouldn't have detected that there was an issue with the laser. And then this is what it looked like after we had it repaired. And this was actually, at the time, a very new microscope, so we were quite surprised to see this. So although we struggle, like a lot of facilities, with having enough staff to do this testing routinely, I would suggest these things should be done, especially when you get a new system, when a laser is replaced or repaired, and then maybe once a month would be a good timeline to aim for. And this same testing can be done on LED or mercury-based light sources on wide field microscopes as well. So I just wanted to bring your attention. I know you've had other speakers talk about core-op-LEMI, but this is the quantitative quality assessment and reproducibility for instruments and images in light microscopy. And it's a large international group now that's really grown quite quickly. And they've put out a new protocol now, which is available on Protocols.io for measuring illumination power and stability. I'll be honest, I haven't read it in detail, but It is Protocols.io is a really interesting website. It gives you the ability to read, to comment, and then the working group, which is an international group who's come up with this standard, can see the comments. They can decide if they want to modify the protocol, and the Protocols.io keeps an archive of all the changes. So it's really a great resource, and this group is the first of the Core Limi groups to publish a protocol. So I would recommend taking a look at that as well. So the next thing I'm going to spend a bit of time on is resolution. So how do we measure the resolution of the microscope? And probably the most commonly used method is using a point spread function. And this was another project with the ABRF Light Microscopy Research Group where we looked at 100 nanometer diameter fluorescent microspheres. And you can see an image here on the left of a sample. And we have a detailed protocol in this paper explaining how to prepare these samples. What you'll notice is the spots are different brightness. So it's most likely that the dimmest ones are single microspheres, and then the other ones are probably aggregates, which is why they're looking brighter. So in this study, we actually selected individual microspheres. We had 3D image stacks, and we used a tool called Psf J and Image J, Fiji, to measure the point spread function of individual microspheres. And I think the next slide, we measure the full width half maximum in the lateral direction and also in the axial direction. So we get the XY resolution and also the actual resolution. And as I mentioned, we use this PSFJ plugin. So this is a bit tedious because you're doing one bead at a time. And I'll show you in our latest ABRF study, we have some code that was written for MATLAB that will look at all the microspheres in the whole data set, but then you just have to be aware that you may be getting doublets or triplets of bead aggregates. So in this paper, we also talk a lot about being able to look at aberrations. So you can measure the resolution, but you can also look at the shape of the point spread function, and you can find all kinds of different artifacts, which could be something to do with the sample preparation. It could be a problem with the objective. And so you can see some of the examples in the table here. There's one here showing coma, where the point spread function looks almost like a banana, and that's usually a damaged lens. So that's something that has to be sent back to the manufacturer, either repaired or replaced. You can see things like a tilt, which is usually the stage drifting during the acquisition. If you have a DIC optics in place, you can see a doublet of the microspheres due to the DIC prism. And then one really interesting thing we found in this study was these really elongated PSFs. And it was normally people using water immersion lenses, but not adjusting the correction collar properly. So they were getting these point spread functions that were about 2 microns in depth, even though it was a high NA lens. So you can really see a lot just by looking at the shapes as well, not just actually fitting the point spread functions. So there's a couple of other tools out there. And like I said, we use the fluorescent beads, but it can be a little tricky to prepare those. So there are some commercial options. This is the Argo Light standard slide, which is multi-purpose. It has a number of different patterns on it, and I'll mention a few of them as I go through the presentation. But they are about $7,500 US. So usually I recommend to people to put it in an instrument budget if they're writing an instrument grant. And we actually just purchased one as part of a new instrument, so I'm looking forward to trying it out. And the way that they do resolution is they have this pattern here where you have these 2 widely spaced lines that are consistent, and then the 2 lines in the middle get closer and closer together as you move down through the pattern. And they have some nice software that comes with the product. So you can see here the lines are getting closer and closer together as we go to the left. And then they have a nice software that will find the peaks and the valleys automatically, and then determine where those 2 peaks turn into a singlet, and you can no longer resolve them. So you can really put this on and automatically measure the resolution of your instrument. Another nice option is the Psf Check Slide, which came out of work by Alex Corbett at Exeter University. And this is using laser writing, and it's quite a bit more affordable. It's $500 US. And he has a lot of similar patterns and so on for doing different tests. So there is a nice laser written grid pattern. So each of these is a point. So you can also do your point spread function. And I guess the advantage over the fluorescent beads is this is reproducible. It's the same sample every time. It's meant to last, I think, pretty indefinitely because it's laser writing in the fluorescent side. And the really nice thing about this is he also has it in 3D. So you can see this is a line of spots in the array in XY, and in XZ you can see that they actually have multiple rows of, or multiple layers of the spacing. So you can actually measure like a micrometer in Z as well. So these are 10 microns apart. And I don't think I put this slide in, but we actually were testing this on our spinning disk, and our point spread functions look like little flat disks. And we realized that the piezo calibration was off. And so anybody doing Z measurements would have been getting really artifactual numbers out of it. So this is a really nice feature for both X, Y, and Z resolution, as well as testing your motorized state, your piezo stage, or the motorized components within the microscope stand itself. And the last one I'll mention is GataQuant. This is a company that sells DNA origami slides, and they call them nano rulers. And they have them developed for all different techniques. They have sim, stead, regular confocal, with different colors. So like the stead would have dyes that are known to be good for stead. The confocal ones are available in multiple colors. And they're little rods of DNA which have dyes on them at a very fixed distance. And this is an example here showing the 90 nanometer steads with confocal and then with stead. And you can see this looks like 4 spots here, and you can see the doublets when you have the stead imaging on. And these are about $350 to $400 each, but the downside is that they're only stable for a few months. So after a few months, you might lose one of the dies on the ruler. So the ruler's not very good if you only have 1 spot instead of 2. So I don't know if they've, haven't kept up on their products recently. I don't know if they developed ways to keep them more stable, but it's a bit expensive if you have to replace it every 3 to 6 months. So next I'm going to talk a little bit about field uniformity. So in any microscope, we know the optics are not flat, and so the lenses do a lot of corrections, but we still usually end up with shading across our images in both bright field and fluorescence. So this is an example of a kidney section stained with H&E, and you can see that there's some shading. This corner and this corner, the corners I guess are kind of dark. It's kind of bright over in the middle to the left. And this is a shading image, and basically you just go out of focus on the sample, or you take the sample off and take a picture, and then you can do a division and correct for that shading. So for bright field, it's not as critical because you're normally counting things rather than actually measuring intensities, but it may be important if you're doing stitching or looking at a large sample and need to avoid those shading artifacts. For fluorescence imaging, it's more important because that shading also affects the intensities if you're doing intensity-based imaging. So normally we'll use the fluorescence slide in order to take an image of the illumination pattern across the field of view. And then you can do the same thing by dividing the sample by the image and get a nice flat field correction. And this is just showing the same data with a colored lookup table, so you can really see how drastically different the intensities are before and after the correction. And this is an example just showing you also need to correct for background. So if you want to measure intensities and you have a high background, you also have to correct that. So it's important you correct for the shading first, and then you choose a background area where there's no signal and correct for the background. And here I just show a table showing you how important this is. In this case, the background of the image was quite high. So if we look at the raw data, cell 1, 2, and 3 actually all have very similar intensities. So this would be looking at this image here before we do any of the corrections. And they're really not that different because the background intensity is really high. After we do the shading correction, you can see that the differences are quite a bit more. But then when we fully correct by doing shading and background correction, you can see that cell 3 is actually about 4 times brighter than cell 1, whereas with the raw data, it was only showing up as 30% brighter. So it's really important that people understand and know how important it is to correct both for shading and for background. You can do some controls and see, like on the laser scanning confocal, if you zoom in a little bit, you may not actually have to correct for shading. So you can just do some tests and see. We normally say if the corner-to-corner intensity difference is less than 10%, you're probably okay to skip that step. But it's actually pretty straightforward to do, so it is recommended. So if you look back to the Argo Light slide, they have a 2D array of rings. So each of these spots is a small ring, and you can use that also to look at field uniformity. So you can look at the intensity of each of these rings across the field of view. And then they use the pattern for a second test, which is for distortion in the field. So if you have, if the mapping of the signal onto the camera or onto the PMT is not perfect, then you can actually measure the displacement of the spots based on a theoretical grid of where they should be. And in their software, they also have this field distortion map. And I believe they have ways to then take that back and fix the distortion in your image. Similarly, on the Psf Check Slide, they have a grid pattern. So rather than spots, it's a full grid. And you can see it here, image at high resolution at 63 and here at 20x. And then if we look corner to corner, you can look at how the intensity of the peaks as you go across the grid pattern change. And you can see with the 20x, you can see this nice pattern that you could then correct for. So you basically can measure the field uniformity and correct for it. They also suggest you can use a large low-pass filter in order to remove the grid pattern and really smooth this out so it looks more like the type of image you would get with a fluorescence slide. So next I'm going to talk about linearity of the detector. So this is one where it's really important to see if your photomultiplier tube or your camera is responding. If you have twice the intensity, do you actually get twice the signal on your detector? And this is something that should be checked relatively regularly as well. And what the Argo Light slide has is variable intensity squares. So they make these squiggly lines on the surface going from an intensity of 1 to 16. And you can see in their software, they can also identify those structures and measure the intensity. And you can see it's quite linear. There's a little bit of deviation in the low and high intensity range, but quite minimal. The Psf check has a similar type of pattern. It's rectangles that are across the sample and going from bright to dim. Those are the two I know of. I don't know of anything more affordable like the plastic slide or something for this. I do think there's I think there's a way you can do it with your cell phone by changing the brightness on the cell phone, but it's really important that the signal going in is really well characterized, and so it's not so simple to create something different than this, like something that would be really affordable and easy to make on your own. So these two are good for that. So color registration. So we know if you're doing multicolor imaging, if you have any shifts between the channels, it's going to really affect your measurements of overlap, colocalization, co-registration is really important. So these are a few examples. This is showing tetraspec microspheres, which normally come in blue, green, orange, and red. And you can also use the larger ones, like the 0.1 or 100 nanometer diameter ones are quite dim. And so it can be quite hard to find them on the slide and image them. So you can use larger ones and then just look at the edge of the microspheres and align the edges rather than aligning the center. So I believe these are the 100 nanometer ones, but you could use 1 micron or even 2.5 micron ones, and then just look to align the edges. The Argo Light, this is the same array pattern of rings that I showed earlier, and you can just see in the corners here and here, you see some shift between the green and the red, and then this is showing that same kind of displacement map, if you have distortions and not just shifts in X and Y. So shifts are relatively easy to correct for. If it's in X and Y, you just shift one image relative to the other, and you lose a bit of data around the edges of the image, but it's pretty straightforward. Distortions are more complex to fix. This one here is done using a algorithm in Fiji Image J, and then I showed you earlier the Argo Light also has a distortion measurement in their software. The Psf Check has a grid pattern that shows up across all the different colors, and so this one is quite straightforward to image and then align the grids. So the last thing I want to talk about is 3D standards and metrics. So maybe before I go on, I already mentioned the pattern on the Psf Check. So it has the 3D pattern array of dots, and then there's also on the Argo Light there's a 3D pattern, a sphere that you can also use. It's a large sphere that you can use for 3D measurements. But we aimed with the Light Microscopy Research Group at the ABRF to set up a sample that was pretty straightforward to prepare, affordable, and easy for people to use. So we developed this 3D standard and we sent it to users all around the world. We had 15 countries, hundreds of data sets, and really covered all instrument manufacturers and types. And you can see a summary here of the different countries that were involved. And you can see that we really have representation from all the major manufacturers. I was talking a little bit with Lionel before we started the webinar, and we actually started this study about 10 years ago, and we probably spent about 3 years just in the sample development. We tried a whole bunch of different things that some were working, some weren't working, and so it took us a while to settle on a sample, and then we actually sent them out to everybody and had to get the data back and analyze it. And because everybody was doing this as a side project in their free time, it's been really long. But I think you'll see the data is quite interesting. But the reason I mention it is you'll notice here that the microscope types don't include the more recent confocals. So this was all done on laser scanning confocals. But again, the sample would be useful for any type of microscope. So what we came up with was using a product called Sigel. So it's shown here, and it's from a company called Biostatus based in the UK. It has a refractive index of 1.365, which is pretty close to what you would have in tissue. And it's a really interesting product. It has it's a liquid when it's cold, and then when you warm it up, it solidifies into a gel. So we use this to make 100 micron thick samples with microspheres embedded in it. And so one set of samples had the small 100 nanometer microspheres for measuring resolution and signal to noise. And then we had a second set that had 2.5 micron microspheres that were different intensities. And we sent the kits to labs to make the samples, and we also sent detailed protocols for sample preparation and image acquisition. And then the participants sent the data back for analysis by the Light Microscopy Research Group. And I have a link here to the YouTube video on sample prep, and then there's a link here to GitHub, which shows all of the protocols and procedures, the spec sheets for the fluorescent microspheres we used, and so on. So one interesting thing, one of the reasons it took us so long is we prepared our first set of samples, and we spent a full day preparing samples, and we sent them out to participants all across North America and globally, not thinking about our Sigel product, which I told you is a liquid in the cold. And so when it went on the airplane in the cargo hull, the sample cooled down and all the microspheres sunk to the surface of the cover slip or the slide, whichever way the slide was oriented, and the samples were unusable. So we had to go back to the drawing board, and then we decided to come up with the sample kit, which we sent out. And then we were trying to minimize the error from sample prep. But this way we had to have each user make a sample. So we spent the time to make videos and protocols and make sure everybody was making them in the same way. So as I mentioned, we had one sample that was 100 nanometer green microspheres for resolution measurements. And that sample also included 1 micron diameter orange microspheres to help people find the cover slip and focus on the surface of the sample. And then the second sample had dim and bright green or dim and bright red 2.5 micron microspheres. They were also sent microscope slides with an EM spacer that was already stuck on the slide. And then they would put the side gel into this chamber, so it would be 100 microns thick. And then once the gel was cured, they would take off the cover on the sticky spacer and stick the cover slip down. So there you go. So this is an example of what the sub-resolution beads look like. You can see they're brighter at the surface due to the fact that there's going to be less scattering and less spherical aberrations, and then you can see they go throughout the whole gel, but they are a bit dimmer as we go deeper in. This is about 100 micron volume here, 100 micron deep volume. And this data here was collected with an oil immersion lens. So if you remember, the side gel refractive index is about 1.365 and oil immersion is 1.515. So you're really seeing the spherical aberrations also causing the point spread functions to elongate and also causing loss of intensity. So I'm going to step through some of the data now. So this is data broken down by quadrant. So what we did is we broke the data into 0 to 25 microns from the surface, 25 to 50, 50 to 75, and 75 to 100. And then we looked at the lateral and the axial resolution as a function of depth. And then what you'll see here is it's broken down into the numerical apertures of the different lenses. So you'll notice for the lateral resolution, the 0.7 NA is pretty consistent across the whole depth. And this is an error lens and likely not being affected as much by spherical aberration because of the lower resolution. The 1.2 NA lenses were water immersion, so you can also see that there's really no effect of resolution with depth. But then when you get into the 1.25, 1.3, and 1.4, you can see that the resolution decreases, so the width of the PSF increases as we go deeper because of spherical aberrations and that refractive index mismatch. We see the same thing with axial resolution, except it's much more pronounced. So you really see a huge difference in axial resolution as we go into the sample. And again, that's due to the spherical aberration causing distortion in the point spread function. And you'll see with the water immersion lenses, it's actually pretty flat. We think the fact it goes down in some cases is probably due, again, to maybe not having the correction collar completely adjusted, so that the ideal resolution is deeper into the sample than you would expect if it was properly adjusted. So there's a ton of data here, thousands and thousands and thousands of microspheres. Thomas Pengo from Wisconsin wrote a MATLAB script to automatically find and fit all of the point spread functions. So there's data from hundreds of data sets and hundreds of beads in each data set. So this is a figure I really like. It's got all of the beads from all of the samples from the different NA categories, and then they're color-coded by quadrant. So quadrant 1 is blue, quadrant 2 is red, and so on. And you can see here in the lateral resolution that it's pretty even across the violin plot. So you're seeing data from all different quadrants is pretty similar. But really interesting when you look at the axial resolution and you go to these higher NA lenses where we know the index of refraction is a bit mismatched, you can really see the difference here where the quadrant 1 has much better resolution and then quadrant 4 is much lower. And so it's really nice to see the richness of this data set and how much information is in there. So the second part of the study was looking at variable intensity microspheres. And we wanted to be able to look at how to quantify the intensity in a thick sample. So we had two sets of microspheres. We had green microspheres that were either 35% or 3.7% intensity, so a ratio of about 9.5. And we had red microspheres that were 40 and 4.7, or a ratio of 8.5. And the idea was to look at those intensities as a function of depth and use it as a way to calibrate the microscope and also validate the intensity measurements. So you can see here an example of the green microspheres where you can see the dimmer ones and then the brighter ones in the image. And then this is just a 3D image from Amaris showing both the bright and dim red and the bright and dim green microspheres. And this is an example from a 63x 1.4 immersion lens. And you can see the different microspheres again. We see dim and bright, and this is as a function of depth. And then we use the surfaces feature using the spots counter in Amaris. And you can see that we're able to identify all of these microspheres. You can also see this example here where we're looking at the just a sampling of different microspheres as a function of depth. And you can see these are the green microspheres that as we go down into the sample, so down to quadrant 4, that similar to the resolution beads, we see this loss in intensity, and we see a bit of a broadening of the point spread function, again, due to spherical aberrations. So it's really important that we're not, if somebody's using this kind of a sample, or if they're looking at tissue that's really thick, that you're not comparing intensities near the cover slip to intensities further down in the sample, because you have this intensity loss. But you could use a sample like this to calibrate the intensity loss over depth and then correct your data for this for these spherical aberrations and scattering. So the most exciting thing to me about this whole study was that we looked at the ratio of the microspheres, again broken down by quadrant, and across all objective lenses, all microscopes, all researchers, we had about 100 data sets from 47 different confocal laser scanning microscopes, 20x to 63x magnification, 0.7 to 1.4 NA. And the ratio that we get of the green microspheres or the red microspheres is really, really robust. So coming from any of those labs, any of those sample preps and so on, we get that really tight distribution. There are some outliers for sure. These could be errors in segmentation. They could be that that lens in particular had some issues. There's lots of different things, but you can really see that the majority of the ratios are coming up just as we would expect. And so it's showing also that you can really compare intensities if you stay within like the same quadrant or a similar depth within the sample. So we really think this is a really robust new standard to really make sure your microscope is quantitative. If you want to calibrate it so that you can correct for the intensity loss and resolution losses as a function of depth, you can also do that. So I want to wrap up with just some ways you can get involved if you're interested in this topic and you want to be part of the community, or you want to keep track of what the community is up to. The ABRF Light Microscopy Research Group is doing another study right now. They're looking at reproducibility of image analysis. And so the title of the project is 3D Image Analysis Tools and Reproducibility. And they've provided two different types of data sets. One is synthetic images of nuclei, and the other is synthetic images of fish fluorescence in situ hybridization staining in a C. elegans. And then they have some calibration images. And they're asking people to download these images, segment the objects, do some measurements like measuring intensity, volume, location within the image, send the data back to the group, and then they want to compare the results that different people get from different labs. And they give minimal instructions on how to segment and measure because they want people to use the protocols that they're already using in their work. And then they ask you to also send a detailed protocol of how you did the analysis. So this is ongoing, and I think you can still sign up for that. So the link is also here on the slide. Core LIMI has 13 working groups now, and many of them are titled the same topics that I was speaking about today. So illumination power, field uniformity, lateral axial resolution. They're all working on protocols to really standardize how everybody is doing this globally, and to eventually collect all of this type of data as well, and use that for higher order data analysis to really understand where, say, resolution can be a challenge in different microscopes, and is there a way we can improve that. The Bioimaging North America group is setting up a metrology suitcase program, and this is based on a program that was developed by the French multidimensional fluorescence microscopy group. And we've decided to start with a relatively basic kit, but the idea is to send the kit around to different labs, so they'll have the tools as well as the protocols in order to quality control their microscopes. So it includes the Thorlabs power meter, the slide-based one, Psf Check Slide, and Tetraspec Microsphere slide. And the idea is we'll also eventually have a database where you can upload all of the data, so that we can collect data. You can look at microscopes over time. You can look at comparisons of the same microscope in different locations and so on. And right now there's 3 cases, one in Wisconsin, one on the west coast of the US, and one in Montreal. And those groups are prototyping them now to really make sure the protocols are clear, the logistics of moving the cases around are well developed. And then they're planning 2 more cases, one for the east coast of the US and one for Mexico. And yeah, the idea is the kit will build. Eventually, we hope to add an Argo Light slide, but we wanted to do this pilot first and see how things go. So what's next? So I just wanted to kind of have some maybe take-homes and sort of what I would like to see in the community as well is I think we need to continue to raise awareness. I know a lot of the users in my facility, they maybe aren't measuring quantitatively at all, or they're measuring and not realizing there's artifacts in their data. So I think we have to convince institutions and funders that this is important, and for them to invest in the quality management of microscopes. So there is a cost to that, of the imaging scientist's time and the instrument time. So I think quality management needs to be a key part of facility budgets. And then researchers and journals need to be convinced of the importance of these quality measurements, so that they can have robust, reproducible science. And then I think Core is really tackling the next to develop robust, community agreed upon standards and protocols. And then ideally, these would be automated as much as possible. So I put here at the bottom, my dream would be that you go to the microscope, you put your standard sample on, you go and get a coffee, and then while you're having your coffee, the microscope is imaging the sample, running the analysis, and you come back to either, yes, everything's great, or there's a problem that needs to be addressed. And that can be logged and linked to your image data, so that, you know, a reviewer or a collaborator would know that that data was collected when the microscope was really in good shape and good condition. So this work was, a lot of this work was done with the Advanced Bioimaging Facility, with staff, former and some present staff. And then I listed here the ABRF LMRG members who were involved with the 3D bioimaging project. And then you can see this is BINA and Core have been really involved, and then funding for my own research lab and facility from the National Sciences and Engineering Research Council of Canada, and the Research for Nature and Natural and Technology Research of Quebec. And thank you for your attention, and I'm happy to take any questions.
Thank you very much, Claire. Great talk. There are some applause here.
Thank you.
So thank you very much. It was a really nutritive talk with a lot of information. I took a lot of notes, and I will reach you back for some details of some of these slides for calibration that I wasn't aware. and I think it's really, really important. Let me see if there are questions. Well, for people in the audience, if you want to open your mic, you can do it and do the question by your own to Claire. If you don't feel comfortable with the language, you can also do it in Spanish, and we will translate both directions. Or you can, write in the chat, and I will read the question to Claire. So I have to begin with, I have a couple of comments and questions, Claire. One that I have is you already answered during the presentation, is how often you do, in your facility, or you recommend in your facility, to do this laser stability, because this is something that probably for most of the users, they will not be aware of this kind of change. And it's our work to have the people aware of, changes in, as you mentioned, right? Replace of a laser, I don't know, replace of a lamp, or either basically the death of a laser over time, which is always something that, well, is what is going to happen, regardless of what we can do or not. So you mentioned once in a month. This is what you more or less do, and basically you run measurement over the different time scale you suggest, right?
So I'll be fully honest that between busy microscopes and being understaffed, we don't run it as often as I would like. We tend to run them when there's a problem, right?
Yeah.
I do think once a month would be ideal. Even better would be to have it, like I say, you come in the morning and just QC the microscope at the beginning of the day.
Okay.
I think for the longer term ones, it's not so practical to run them every day, but certainly for the seconds to minutes kind of tests, you could easily run that over the course of a half an hour or something like that, right?
Yeah, I wasn't aware about this Psf Check Slide, which
Mm-hmm.
is seems to
Yeah.
me very, very useful, because
Yep.
always preparing these beats is
Yep.
sometimes difficult, because you need to sonicate, you need to have
Yep.
a good
It is tricky.
medium
Yeah.
in order to have a in good condition, right, to run the experiment. So it's
Yep.
very nice. This one question is, this slide, PSF check slide, is the same for all the different measurements that you mentioned, the PSF as well as the uniformity of the illumination?
Yeah. So all those patterns are on a single slide.
Oh, nice.
And the slide itself is green, but I think you saw from the example I showed, it does seem to work across different colors. And I haven't checked with Alex Corbett recently, but I know he was also working on materials with a different refractive index.
Nice.
So right now it's the plastic slide from Chroma. So I think it's pretty similar to oil actually. It's pretty high refractive index, but he's trying to make ones that are, closer to water, and so you could have different
And this is
different slides
commercial.
depending on. Yeah, so you can. I'm happy to share the slides. The links are all there, so you can just go to their website
Okay.
and order the slide. Yeah.
And regarding to your preparation, the one that you use for the 3D standard, is this also well? I understand that basically the beads and the medium is commercial, but are you preparing this by we can prepare by our own, or is something that you can also order?
No, you really
Whole
need to
set, I mean.
prepare. Yeah, you need to prepare them fresh. They last for maybe a week or two because the side gel dries out over time.
Okay.
So even if you set it with nail polish, it still dries. So we found 1 to 2 weeks is okay. And you also have to make sure you don't put it in the fridge because
Yeah.
and your beads will all settle. So it's best to prepare it fresh. But the nice thing is the side gel is a liquid in the cold, so you could keep a side gel bead mixture in the fridge and then just vortex it maybe sonicate it and vortex it, and then make a sample, and it hardens almost instantly. You could almost prepare it right on the microscope.
Ah, nice.
So there, it's easy to replicate that way.
Okay, great.
Hmm.
There is hands right by Daniel. Daniel, do you want to open your mic?
Oh, yes, thank you, and thank you, Claire, for your nice talk. I have a question about your the last part of your talk.
Yeah.
When you show these quadrant-aware normalization, let's say, where you corrected your intensity, which was fading by depth, let's say.
Hmm. Yeah.
You may have said it, but I may have missed it. How is that you correct this fading effect?
So we were focused in the paper more about measuring the resolution and these intensity ratios. We didn't do corrections, but you could measure the change in intensity with depth, and you don't have to necessarily go to quadrants. You could do it more refined, right? Because you have the exact position of each microsphere, and then it would be almost like a detrending, right? So you would remove that trend from your data. So for instance, similar to a shading correction, really, that you would divide through your data and get a fixture intensities that way. Yeah, so you could, depending on, you need enough microspheres. That was one thing. When we have the paper ready to publish, there's a lot of detail about filtering the data, if there were saturated pixels, or sometimes we didn't have enough microspheres in the data set to do the ratios. So you do have to play around a little bit with the bead density, I guess. But it should be a relatively straightforward correction.
Okay, thank you so much.
Welcome.
Any other question from audience? I don't see anything in the chat. Grace. Claire, hi. Can you hear me well?
Yes.
I'm wondering, do you think this is just kind of more of a philosophical question? Do you think we're going to have sometimes some kind of quality control test of instruments that would be required for reporting results from that instrument or something like that?
Yeah, I mean, like I had sort of said on my dream slide, the
Like because
ideal.
you put in your slide something that's certified, like
Yeah.
some kind
Yeah.
of requirement of
Yeah.
okay, you're publishing with this instrument and then you're a certified calibrated instrument.
Yeah, and I think ideally this would be built right into the microscope manufacturer software, right? So you put the Argo Light slide on, or you put the Psf slide on, and the software would know where the different patterns were, right? So you could have it set up so it knows where the 3D array of points is, and it knows where the intensity calibration is. And then, like I say, you should be able to just hit a button and say run calibrations, right? And this is what happens on a lot of other fields in MRI and get in mass spec and flow cytometry, right? And
Do you think we're
then,
going to see that ever in microscopy?
well, to be honest with you, I've been working in this area for close to 20 years now, and it's always been very grassroots, and all these studies by the Light Microscopy Research Group, as I mentioned, we're kind of doing in our free time. But I think this core app community has really changed the game. And I find it very satisfying because I'm also very interested in networks and working together and moving together as a field. And I think that in a very short time, it's really clear that the manufacturers, the journals, and soon I think the funders are going to see, the power of this organization. And I know Katerina Stramboy Castilla was telling me she's approached the camera manufacturers now about getting all the metadata about the cameras into the images. And she said because she approached them as core app and not just as one researcher who's interested in this, they're all on board and they're all working with them to do this. So I think we have that momentum with the international community, and I think the people who are in the community already knew how important it was, but now I think because we formed this big global group that the manufacturers and the journals and these groups are going to pay more attention because we have that sort of momentum of the whole community, you know. So I'm optimistic that we'll see a lot of change in the next 5 to 10 years.
Thank you. Yeah, I was, we already have discussed with Caterina about this suitcase and other things related to calibration, because this is something that I would really like to approach through our LABI, our Latin American Bioimaging.
Yeah.
Because of course these are expensive tools, right? And it's not always simple. You know, as you mentioned, probably it will be recommendable that when you get a new microscope, you get also some control tools, or you know, samples, calibration tools, in order to make well, more feasible to make it work in long term. But it's not that simple, I mean, in the region. And I think it's something that we can do together. Probably if we approach Torlab and many other of these other companies, and we say, well, we are a whole community, we will put some money together, they will also help.
Yeah, I'm happy to chat more about the BINA program. They were able to get really deep discounts on all the components because it was the community reaching out, and we're talking about maybe putting the Argo Light into the kit so that people can use that without having to own one, you know. Exactly. And then from my facility, we just put in a purchase order for the Zeiss lattice light sheet, and I was able to get an Argo Light slide as part of that purchase. So I'm hoping, because this stuff is so difficult, like I said, to like on our workhorse equipment, it's really hard to justify the time, both the instrument time and the staff time, to really calibrate them. But I'm going to have a full-time person working on our lattice light sheet. So I'm looking to use it as a bit of a prototype and see if I can push Zeiss as well to maybe build some of these tests right into the instrument, you know, because it's a new instrument, maybe they'll be more interested in developing it. And then just one other comment is they have a lot of these tools already. Their engineers have them, the software as well as the devices. Like they have a way of measuring the laser power. They have a little photodiode in the system to measure the laser power, but they don't report it to us, you know?
Yeah.
I know that Zeiss Elira has a PSF measurement tool, but it's hidden. It's only for the engineers. So I think there's a lot of things out there. We just need to get the will, I guess, of the companies. I guess they want their instruments to be performing well, but they also probably don't want to know about repairs that we didn't notice. And
like absolutely.
No, I was just going to ask you if that project that you, because I didn't understand if it was a BINA project, that the one that you're sending the calibration
with the suitcase. Yeah, so that is a BINA project. Yeah, okay. So it's for Mexico, the U.S., and Canada. But we'd be happy to chat if you're interested in doing something similar. We learned about it from the French group, right? So yeah.
Good. And I remember in the presentation of this guy from France in the
Orestes,
in January for the Royal Microscopy Society. The suitcase was really overload of different kind of calibration tools and different apparatus, etc. It was really expensive. It was like 30,000, something like that, the suitcase. But this is not that expensive.
Well, but even I mean, if it's a project for 7-8 countries, yes.
So we decided we wanted to go very simple, and we thought the two most important ones are your stability of the illumination and your resolution, with the idea that we can build on it. So we bought, instead of having one really expensive kit, and the other thing is if you have a kit that will measure 20 things, you have to do 20 things. So the kit has to stay in your lab for a month or 2 months. So we thought with this simple kit, you can probably get all the testing done in a week, and then it could go to another lab. And then we could decide, you know, or maybe you have different kits. You have the basic kit and the advanced kit. And so the one we have, I can talk to you offline, but I think it was only a couple of thousand.
Yeah,
And then our idea right now is to keep it in keep them in major centers. So we have one in Montreal, and we thought we'll just walk it around, like meet
Yeah.
for coffee and hand it around, and then we'll ship it to Toronto and have the Toronto labs use it, and then, you know. So I think there's it's been really great working with Bina on that to kind of design a really nice project, and then we can build from there, you know.
Yeah. Great. Any other question? I don't see anything in the chat. So if there is no more question, we are in the hour. So I really want to thank Claire for the seminar today. We are really excited with this series of seminar. We are learning a lot about how we can support quality and reproducibility to our users, and also for us, right? For our group. So thank you very much, Claire.